Welcome to biology. We're continuing with our conversation around DNA and really how DNA will begin to form and code for proteins. And so you might recall from our learning target I have here at the top, it is really called DNA to protein. Construct an explanation based on evidence for how the structure of DNA determines the structure of proteins, which of course we know can carry out essential functions of life through systems of being specialized, right? So every protein can have a very specific function. Um, so with that, we already talked about really the process of protein synthesis, and so as a review, you can click and drag these in order of the events that they occur. And the biggest hint I want to give you is that uh, in our previous activity, we talked about protein synthesis, and really that is broken into two parts. And so within those two parts, you kind of have a few steps, but the parts were transcription and then translation. And so I kind of tried to help you out at the end of the descriptions there. Then, with this, and as we focus more on, well, how does our genetic code create these kind of customized proteins, if you will? And to understand that better, we really need to look at genes. And so, got the formal definition here, but in biology, a gene is a basic unit of heredity, right? That means we can get it from our parents, or it gets passed on through our DNA, and a sequence of nucleotides in DNA or RNA that encodes the synthesis of a gene product, which is RNA or protein. So it's really a portion of our DNA, not the entire DNA. And so these segments then code for very specific things. And so altogether, it can help create us, right? So to help with this and to talk about that further, I included this video. It is super helpful, super visual, and I really want you to watch this to help you have a better understanding of how our genes will create these proteins that are very specific. So that being said, a couple key points that I really want you to pull from the video I wrote down here. So first, every gene is a sequence of DNA that will create a specific protein with a specific function. And then based on the sequence of amino acids found in the gene, they will potentially form unique shapes with a unique purpose. And so really, we have the various amino acids that we talked about earlier when we introduced proteins. And so we have essential and we have non-essential proteins, or sorry, amino acids. And with that, then, right, every combination of those can create something different. So that's important for us to bring out of that video. With that, okay, I wanted us to look at this codon chart. We practice coding and taking mRNA and creating these amino acid polypeptide chains. And with that, I wanted us to look at, well, how does in our DNA, how does that signal to the mRNA and to the ribosome and so forth? How does that signal uh, tell the gene like when it's over, right? Why doesn't it just keep building one big protein? How does it separate the proteins and so forth within the strand of DNA? And so, looking at this codon sequence chart, I want you to take a look at that. And there's a hint here with the word stop. Okay, so with these codons, again, tRNA is looking for bonding sites to match up, and then it releases these amino acids, or kind of attaches these amino acids, if you will. And there's a few codons in here that don't have an amino acid, but they actually tell it to stop. Whoops, I have that written in there from class. Next, I want us to go through the process again of protein synthesis. And so with that, you have this chart, and so you will see that this first row is filled in for you. That's the original DNA sequence. You then need to do a copy or a complementary strand of the DNA sequence. So remembering our bonding patterns. Then, just like we practiced the other day, I want you to be the RNA polymerase, and I want you to make an mRNA strand that's going to get sent out of the nucleus to the ribosome. And a reminder that RNA has different nitrogenous bases that bond, and really it's just that there's no thymine. Excuse me. So if our DNA has a thymine, that's fine because that RNA can attach an adenine still. But if our DNA has an adenine, it cannot attach a thymine. It has to attach a uracil. So here's a little example there for you. I did a few snippets here. So again, it really would look like A attaches to T in the DNA, and then T to A in the mRNA, and then trying to create the tRNA. So this is going to be the anticodon. And just a reminder, this is the codon. So when you look at the codon sequence chart, this is the three that you need to look at, but the anticodon needs to bond to it. So the opposite then again in RNA of A would be U. Okay, so there's an example there of how that works. Then you're going to need to use the codon sequence chart. So if you go back out, take a look at this and search for that codon. Again, the mRNA sequence is what this is. Uh, so make sure you get that right. And so then that would be our first amino acid. And you just need to know the little abbreviation, as I don't expect you to memorize the names or anything like that. Last but not least, I have this other video for you. Uh, and really, it's kind of a controversial topic in a way, but it's really the future of kind of this technology that we can modify our genetics. So please watch this, and we can look at how genes can be kind of cut and pasted and moved around. And as we learn more, scientists are able to do more and more. So watch this video, and then this is a short essay regarding the video and regarding our learning. Uh, and so please take your time, especially in this first part. This is specifically from the learning target. So, as my perspective, I can look at this to really help determine, yeah, are they getting the learning target or are they not? So, please make sure you do a good part on this. I gave you a suggested work bank. If you're doing a really good explanation, at least in my head, you should be using these terms from our learning so far. Okay, let me know if you have any questions, and always feel free to email me.